Hello and welcome to our Lakeside chat. We are David Wofford and Katie Noonan, co-chairs of the Rotary Nature Center Friends. And we're so happy that you can join us tonight.
As we begin our program, it is important for us to acknowledge that we are on stolen land, that Lake Merritt is part of Ohlone territory. We hear now from Corinna Gould, spokesperson for the Confederated Villages of LaShawn and co-founder of Segura Te Land Trust.
Good afternoon, relatives. My name is Corinna Gould. I am the spokesperson for the Confederated Villages of LaShawn. We are here today at what most folks think of as Lake Merritt. We are in the territory of Huichin. Huichin is actually a territory that encompasses 6 Bay Area cities: Oakland, Berkeley, Alameda, Emeryville, Albany, and Piedmont. And this was a place of abundance. I'm so happy that people from all walks of life that now come into our territory can enjoy this beautiful place that my ancestors have enjoyed since the beginning of time. My relationship to the land that I have been born to, raising my children and grandchildren here, has been to tell the story, the truth of what happened on this land before other people came here. I'm hoping that as we begin to learn these lessons of fires in California, the pandemic that's happening, that human beings come back to living in reciprocity with the earth.
Thank you to Corinna for that inspiring message, and we hope that we will continue to move closer and closer to that goal. We appreciate all of the knowledge and philosophy coming from the indigenous peoples that live here now and lived here in the past. So we are so happy to have you all here tonight. For those of you, our guests who have not been to a Lakeside chat before, we would like to share a few slides about who we are. So Rotary Nature Center Friends is an independent, all-volunteer 501c3 nonprofit since 2021, providing online and in-person programming for all the people of Oakland. We engage the community in real science and stewardship activities, bringing nature and people together in the heart of downtown Oakland to steward the nation's first wildlife refuge. Our focus of activity is community science and environmental justice. And we encourage you to look at our Facebook's website and our website to see more pictures of what we've been doing. We've gathered together people from various specialties: education, interpretation, science, and educators and artists to help us further our goal. So tonight, we'd like to give a hurrah to the fungi and to a philosophy that is expounded by an organization that we want to give props to in our program today. So just to say that the world depends on fungi because they're the major players in the cycling of minerals and energy around the world, said by the famous biologist E.O. Wilson. And we would like to congratulate iNaturalist for receiving the Heinz Award for the environment recently, and to the local people that we're so familiar with, Kenichi and Dr. Okay, I'll get it right. Okay, so we want to thank them. They've been at Lake Merritt doing iNaturalist, teaching all of us about how to connect to nature using our iPhones and to build knowledge that can be shared with scientists and with the interested community of citizens and people. So we would like to acknowledge and say thank you to to Dr. Jerry Lips and to some of the professors and students from the Cal Maritime Academy that came to on a tour of Lake Merritt Channel with us last weekend. Would like to also all of the people who turned out for the Creek to Bay Day and visited our Rotary Nature Center Friends work site. And this really illustrates our reach out to the community, all groups of people in the community, all ages, to share the joy of exploring Lake Merritt. So at this point, I would like to ask if Dr. Lips is in the room. Are you in the room, Dr. Lips?
Yes. You want me to share?
Actually, I think I can do it.
Okay.
Is this looking okay?
Yeah. Thank you.
All right. So
we'd like to announce our symposium that has been approved by the Geological Society of America for the Cordilleran Section meeting in Sacramento on April 1st through the 5th. Andrew Alden and I, as co-conveners, put this symposium together, and it will cover the geology, geography, biology, and history of Lake Merritt, sponsored by the Rotary Club. The audience for this will be a wide range of geoscientists and science students, but we seek scientists, citizens, and students to speak or poster on any aspect of Lake Merritt. The abstracts for talks are now due and will be open until January 28th. That's the deadline for submitting an abstract. Next slide, please. So some of the topics of interest could be geology, water, a whole bunch of things that you can see here. Invasive species, some of the things that we've seen like fish die-offs, modifications of dredging and harvesting. And as I said, abstracts are due anytime from now until January 28th. Next slide. Here's a few pictures to inspire you in the next slide. To inspire you with ideas to maybe join us in this session of both oral and poster presentations in Sacramento. You could do animals like the couple in the upper part of the slide here on the left, or maybe ancient animals or Ohlone shell mounds. There were 6 of those in the vicinity of the lake, a very important part of the history of the lake. And then there's a picture of the old lake in around 1900. The fish die-off was an important event last August 22nd, I should say. And then there's the channel down there that can be included in this, some organisms on the sea floor, and a couple of good views of events in Lake Merritt, including, again, students associated with Katie and her efforts. So if you're interested in contributing a paper or just joining us in another way, posters or as audience, please contact me at jlips@berkeley.edu and we can talk about it. Thank you very much.
thank you, Jerry. This is an exciting opportunity. I'd like to say that this is being sponsored by Rotary Nature Center Friends. We're delighted this is bringing together the scientific community with the local community, including students at high school age and younger, exploring, collecting credible data, lots of times using iNaturalist. Just shows what an amazing resource Lake Merritt is and how much Rotary Nature Center Friends goals are really pretty exciting, and we're excited that you and Andrew Alden have put this together and given us and the wider community this opportunity. So please contact Dr. Lips if you're interested in presenting, or if you're interested in being involved in some of Rotary Nature Center Friends programs where you participate in data collection and exploration of Lake Merritt. Okay, so thank you. We're going to stop. And now I want to thank all of you for being here tonight. Now, here's what we came for. We're going to introduce our speaker, Damon Tighe. Damon, welcome. Nature Center Friends Lakeside chat number 47. This will be like your third or fourth chat that you've done, and we're so appreciative. Thank you so much, Damon. I appreciate it. I'm turning the podium over to you.
Sounds good. I'm just going to drop a link in the chat. That is the slide deck for tonight. So if there's something you wanted to kind of like hold on to and look at, you can grab that. I'll also drop it again at the end of the night, just so that way you can pick it up there as well. Let me go ahead and share my screen, and we'll jump into tonight's presentation on mushrooms of Lake Merritt. So hit the slideshow button, and we should be good to go. Just a little head shake if everybody can see that. Looks good. All right. So I'll do my background because it's always important to know the biases of your presenters. I am not a formal mycologist. There's very few people that are formal mycologists. There's just not that many places authoring degrees out there, unless you want to study Russ and talk about ag science. I grew up in Calaveras County, which some of you might or might not know, maybe from Mark Twain stories about jumping frogs in a bar. Not too much going on in the county, so I ended up in the Bay Area for college at St. Mary's. Ended up going from St. Mary's to Portland to teach high school for a bit. Ended out in Montana for a while at film school for the Discovery Channel. And then ended up back in the Bay Area working on the Human Genome Project for about a decade with the national labs. So worked on the Human Genome Project and then DNA sequencing projects related to mainly carbon sequestration, carbon cycling. From there, I moved to a biotech company where I helped bring hands-on biotech into classrooms and more or less train educators to figure out how to sequence DNA, extract proteins, and do that type of analysis and things like that. But in my spare time, I'm an amateur mycologist that likes to combine a little bit of that background, the DNA sequencing, with understanding fungal diversity in California. And I really cut my teeth on all of this at Lake Merritt. And so that's why when we go through the slide deck tonight, you're going to see a lot of photographs taken directly from Lake Merritt. Also, we're to talk about mushrooms Lake Merritt, so that would make sense too. We're going to do some basics though to get started with, just to kind of so that everybody's on the same page. So we're going to do a little bit about kind of fungal biology, more or less. Understand what is a mushroom, diversity, what things do you look at for identification. And then we're going to pair that up with one of my favorite tools, which is iNaturalist, and think about how do those aspects that you're looking at for identification, how can you translate those into easy to see things on iNaturalist so that other people can help you basically go, yep, that's definitely what you got. And so that we've got this data from a scientific point of view to understand what's going on with species as our world is in general turmoil. We'll do a little bit of how to do data mining in iNaturalist, and more or less this is kind of how I built most of this slide deck, was because I went back through and mined my old data there. So iNaturalist is a great kind of notebook of modern times if you're a biologist. Then we'll do the species tour of Lake Merritt, and you'll get to see all the crazy weird things that are there. You just need to learn how to find them because they are there usually every year. And then we'll dig a little bit into a bigger project that I'm involved in, where we're sequencing basically the species diversity of fungi in California. And this basically kind of grows out of some of the desires I had for Lake Merritt. So mushrooms, we already covered this if you guys were here for the quiz show prior to the presentation. But where do they line up in the tree of life? Well, fungi, they're a lot more close to us than they are to plants. So they like to eat things like we like to eat things. We don't like to make our own sugars like plants do. It's crazy things. So they're a lot more like us than they are like plants. And we see this if we look at them from a genetics point of view as well, not just how do they consume things in the world. If we think about the things that we actually call mushrooms, this is also covered in the lead-up questions. Very, very good. Mushrooms traditionally are just those things that make big fruiting bodies, and this tends to be in the kind of Basidiomycota branch of the fungal groups. Some of the ascomycetes produce things people will, in common speak call mushrooms, and those are things like morels and some of these bigger cup mushrooms. But from a scientific point of view, mushrooms is really just applied to those basidiomycete fruiting bodies. And then if you go down, you realize that there's a whole bunch of this list that we're not talking about them having mushrooms. That's because majority of fungi don't produce mushrooms. They get their spores out in the world in different ways. We're not going to cover as many of those tonight just because they're a lot harder for me to see with my eye and with the camera. But a handful of those will occasionally make big macro things, but it'll be like 1 or 2 species in each one of those little ladders there. So we think about basidiomycetes, which we're going to spend most of our time with tonight. These are the group of organisms that more or less produce a club-like shaped cell that then produce the spores off of them, and the spores are what's going to more or less lead to the next generation. They can come in all kinds of shapes and sizes, and if you want to learn more about them, there's a really wonderful website that some of the San Francisco State folks built a while back called mycoweb.com/CAF. And this basically has a good portion of our knowledge of local mushrooms listed there with kind of easy to read keys like this one. So this kind of gives you the general shapes of a lot of these basidiomycetes. Everything from the agaricus-like mushrooms that many of us are familiar with gills on the bottom, like the button mushroom you might find at the grocery store, which is also the portobello mushroom, which is also the cremini mushroom. They're all just the same species, just different strains and grown for different times. All the way out to things that like the boletes, these wings have giant pores on them. They look like a sponge pad, out to weird things like stink horns that you can find at Lake Merritt. All of those things are all basidiomycetes. In the ascomycete world, not as quite as much diversity in the kind of fruiting bodies per se. It's mainly kind of things that look like cups, things that look like cups that got folded up really weird, i.e. morels, and then things that you'd have a hard time even recognizing were a fungus. Black weird sticks that stick out of the ground or off of wood. But probably the one that gets most of the love here in the ascomycetes are the ones you can't even find above the ground. You have to dig for them, and those are the truffles. These mushrooms that hide underground and use basically a nice kind of gaseous way to get themselves exhumed by organisms, and then their spores spread. If we think about the life cycle of a mushroom, or more specifically, we should be saying the life cycle of a fungus, of a basidiomycete fungus, it more or less looks like this. We'll start with the piece that everybody knows, the big old mushroom. So we've got more or less here a drawing of an Amanita genera mushroom. If we look in on those gills, what we'd find is we'd find those club-shaped spores, those club-shaped cells, with then 4 spores being born off the top of each one of them. Each one of those spores has just 1 copy of the genome, and it gets basically flung out into the world and hopefully lands someplace that is inhabitable for a spore and for a further fungus. What will happen is, if that's the case, the spore wakes up and then will start to grow out into the substrates around it. When it does this, it can find other basically hyphae at this point that are growing around, or sorry, other hyphae that are growing around, and they can then pair up with each other, more or less. This is sort of like, if we're using the marriage kind of connotation from earlier in tonight, this would more or less be like courting. So these hyphae are courting each other. They're more or less kind of living together in this thing that we call mycelium, and it's way, way later though they're going to have fungal sex. They'll live together for a long period of time, then they'll start to build a hyphal knot, and this is usually happening right now. So in the Bay Area, a lot of our fungi are producing hyphal knots. They're recognizing the change in seasons, getting a little bit less light, a little cooler nights, well maybe not this week, and they'll start to basically put a bunch of this mycelium in a dense little knot. That little knot can then become kind of primordia, basically all the stuff you need to have a mushroom, but just really, really compact and with not much water there. Because as soon as we get that first rain, what's going to happen is that primordia will absorb all this water. All those cells that are very kind of dry and packed right now will basically fill up with water and expand really quickly. And this is what's going to produce more or less your mushroom body. And this is why mushrooms can pop up so quickly. They're kind of like those expanding dinosaur sponges, that you throw in the tub and they blow up really quick. It's a little bit like that, but there's also some differentiation going on too. But more or less, that is our whole kind of life cycle for these organisms. If we're trying to identify basidiomycetes, it's really important to look at the shape of the cap, more or less the big portion of the mushroom that's sticking up in your face. So if this is bell-shaped, is it convex? Is it conical? Does it have more of a funnel shape to it? All of those can help indicate what genus you might be in. And then if you flip it over on the other side, especially for things that have gills, how they attach basically to the stem, or what mycologists like to call a stipe, is also a very useful key as to what genera you might be in. And so those can be everything from free, where the gills don't touch the stem, they just touch the cap, and there's a blank space in between. They could be directly attached to the stem. They could run down the stem, which people call decurrent, or they could be notched. They could go towards the stem and then do a little break like this and then come back up and touch. You can also look at spore color as a way to identify what genera you're in. And so most mushrooms, if you were to take them, just clip off the stem, place it on something and let it sit overnight. It will deposit spores, and those spores will be of a different color. One of the things I love about mycology is that there's still so many low-hanging fruit as far as questions go that we don't know. Why do mushrooms produce different spore colors? A lot of hypotheses out there, but nobody's been able to go through and really do a nice test of these hypotheses. And so this is still, if anybody's looking for a master's project or maybe a PhD, I feel like this is a low-hanging fruit because there's some definite good indications about why you might want to have spore color. A lot of these things that like to hang out in, say, bright grassy zones tend to have really dark spores. Dark spores usually have melanin in them. You might be familiar with melanin as the stuff we occasionally have in our skin too. What's that do? Protects you from usually UV radiation. What's UV radiation not good on? DNA. But at the same time, occasionally we'll find that giant white one there was in the grass at Lake Merritt. And so that's definitely a rule breaker. So why is that one able to make it? We don't know, but a lot of great unanswered questions in the world of mycology. Spore colors can range all the way from white to rust to black to purple. There's even green ones out there. Still waiting for somebody to find a nice blue one. So if somebody comes across one like that, let me know. If you want to get really now technical and be able to put like a species name on something, this oftentimes requires microscopy. And so that's going to be looking at basically these sexual cells. So remember we said that those two hyphae were living together as mycelium, and they weren't kind of going through the deed yet. That deed only happens once you get up into the gills, where you actually have exchange of genetic material, and that's ultimately what's going to give you those spores of new genetics for the next generation. So in the ascomycetes, they have these long sac-like cells, and this is actually where they get their name from. Ascus refers to the idea that this is a long wine sac, and this wine sac cell is filled with spores that will basically rupture and then shoot out into the world. And this is why sometimes if you blow on cup fungi, they'll smoke at you. That's actually just all of those spores being released at once due to a change in air pressure. If we look at the basidiomycetes, now we've got that giant club, and it'll have 4 spores coming off of it. So anybody that's a biologist, this is because you would just have gone through meiosis here, and that'll take your cell and then break it out into 4 with just 1 copy of a genome. If you want to get full nerd, then you basically do your microscopy. You're like, I'm pretty sure I know what I have. And then you do sequencing, and then you're like, now I really don't know what I have anymore because nobody sequences. But sequencing is kind of becoming sort of the benchmark in a lot of contemporary biology, because morphology is great in kind of showing us, hey, this is like this, but DNA does another nice level to it, which is we can use statistical analysis to say how close things are to each other, and it allows us to look at kind of phylogeny based upon changes in sequence. And so we can then make estimations about how long ago were these 2 organisms basically branching off of the same ancestor and things like that. And that's things you really can't do with morphology that easily. So we've got 2 types of techniques we use there, Sanger sequencing and nanopore. We'll dig into those later. Guides for California. These are the 3 best guides you can get. Mushrooms of the Redwood Coast, beautiful photographs, easy to read, which I love. California Mushrooms by the San Francisco State folks. This is also a great book. It covers more of California, including the mountains, and has a little bit more diversity. And then the pocket guide that any beginning mycologist has is All of the Rain Promises and More by David Aurora, who I would say him and Gary Linkoff are the 2 people that basically reinvigorated non-academic mycology in North America. And this is his pocket guide to basically getting started. And it's a hoot. I didn't pick it up for years because the front cover scared me. In fact, I remember like looking at this in Powell's books when I lived in Oregon. It was like, nope, that's just too weird for me. I went on to get a plant book. And then 10 years later, picked this up and I was like, wow, I wish I would have gotten that 10 years ago because it's full of vignettes. It's very helpful, useful guide in the field. Really, really good book. But what I lean into even more these days than my field guides is probably iNaturalist. And so Katie already gave a little shout out to iNat at the beginning. iNaturalist, if you're not familiar, is a platform that more or less works like this. Take a picture of an organism, upload it to a global database where other people can help you identify it. And even more so, they've traded in artificial intelligence in there the last couple of years, and it's also very helpful at getting you further down the taxonomic rungs to understand what you have. But the beauty is that it's a community effort. It's not just, I'm just logging this and I walk away. People are actively there kind of looking at the data, using this to kind of build information about our knowledge of places like Lake Merritt, et cetera. So if we're going to use iNaturalist, which is predominantly a visual tool in order to have your arguments or conversations about what this organism is, you're going to want to trap those attributes that are important that we just talked about. So on those mushrooms, right, we're going to want to get a full picture of the mushroom. We want to get a picture of how those gills attach to the stem. We want to get a picture of the top of the cap. And in some things like amanitas, you want to make sure you got a picture of the base of the mushroom. And so a lot of times, if you look at my iNaturalist observations, I have a ton of photographs there because I just want to make sure I'm covering all the bases. Or you could be a really good photographer, which half the time I'm not, and just get all of those attributes in one glowing photograph like this. So all this, this has all those things we just talked about. Once you have all of those, you upload them to iNaturalist, and what happens is this starts the discussion. People get through and they start telling you, oh, actually, I think this is this. Have you thought about this? It's a really, really great tool to learn and at the same time contribute to the scientific understanding of when and where organisms are. And I think that's one of the huge things about iNaturalist is it's just rebuilding species biogeography maps like what feels like overnight. And we couldn't have done this any other way because there's just not enough grad students out there checking on these organisms to generate this type of data. In order to get those good photographs to go, I want to give you guys a couple tricks. Because especially mushrooms, they love to hang out in dark places, and some of them are very bright. And so if you pull out your cell phone and you try and shoot some of these mushrooms, you just can't seem to get the right texture on them. Like, I can't get a good exposure. So the trick you can do, and I was shown this by, I think it was a 5th grader, you take your cell phone, you're taking a photograph of something, and you just hold down on the screen. And what will happen is you'll see what comes up as an AF or AE lock. And now if you zoom up or down on your screen, you can adjust the exposure. And all of a sudden, this chanterelle, I go from having blown out gills that I can't tell what's there to actually being able to see the gills there on the left. And so this is a really, really helpful tool for any time you're using iNaturalist. The other thing is, majority of biological life is not at our scale. We're kind of big organisms. And so if you really want to see the diversity of life, you got to be able to get down 1 or 2 orders of magnitude. And this is the cheapest way to do it. Clip-on macro lenses. You can pick these up usually for under $1 or at least under $5 these days. Pandemic kind of did a number on them. These just clip over the top of a cell phone. And now I can basically go and look at really, really small stuff. So that mushroom right there on the left, that is the size, or actually smaller than the size of my ring that I'm holding. And that ring is in the center photograph. So you can have an idea of like what type of magnification you get with a really cheap tool. And the trick is, buy the cheapest one out there. The really expensive ones are not that much better than the 50-cent one I bought. So when I go out, this is kind of what my toolkit looks like. I've got my clip-on macro lens. I've got some chemistry, some KOH for doing some tests. I've got scales, and I've got sometimes my slides and a microscope if I want to do quick checks, and then a tackle box to take things home and look a little closer. For iNaturalist, what I'll do is I'll take my photographs usually in the field by hitting the observe button. I can then just pull them in from my library on my phone, and then I'll click on what did you see. If I give it a couple seconds, iNaturalist will basically give me a suggestion on what it thinks I have. And originally when this came on, it was just okay. And now it's scary good. I used to upload mushrooms and it would say it was a diatom of some sort. And now I would say in California, probably like greater than 70% of the times for fungi, if you can take a decent photograph, that's got you in the right genus, which is really, really nice, because now this makes it easy for me to go back to my field guides and see what's there. And so let's do a little bit of data mining with iNat, and then we will jump into the species list. So you can do this on your cell phone. You can basically open up the iNaturalist app, hit the Explore button, and then basically look for where you're at. If you hit the little search button here, you can then type in the species you're interested in. So maybe it's just fungi. All of them come up for an area and you can walk right to them. So Lake Merritt, we're going to look at Lake Merritt for doing a quick data dive through the online browser of iNaturalist, which is much easier to use than the phone. So if I go to Lake Merritt on iNaturalist, as of this morning, we have 1,800 mushroom observations, over 230 species. And so we can see Katie's been out there recently putting stuff up, and then Jet has also got some down here. He's got some nice white molds on leaves. And so all of these are up there for your viewing pleasure. So let's dive into some of the more notable fungi of Lake Merritt. So one of the ones that anytime I show people pictures of it, they're like, whoa, where did you see that? And I tell them Lake Merritt, they're like, no way. This is Clathus ruber, the latticed stinkhorn. And this is one that oftentimes you can smell before you can find it. And that is because this is a very unique mushroom in that it doesn't use necessarily wind to spread its spores, it uses flies to spread its spores. So it makes this giant geodesic dome that smells like a rotting carcass, looks like a rotting carcass, and tricks the flies into thinking rotting carcass. They show up, they climb all the way through it, looking for their food. And when they get in the process stage, you can see there's little kind of brown slimy things on there. That's what we call the gleba. The gleba is basically where the spores are being produced in this fungus, and the flies will get it on them. They'll also eat the gleba. The spores can pass through their digestive system, and what are flies good at doing? Flying. And so they will take that spore and bring it to probably a bigger distance than the wind could have taken these big spores. And that's how this one gets spread around. This is not a native fungus to California. This is originally from the Mediterranean. It most likely got here on some nursery stock. But luckily, this is one of those ones that has not figured out how to make a living in California outside of our urban landscaping. So you'll only find this urban landscaping with wood chips. Also, people eat this mushroom. I've eaten it once. Once was enough. Omphalotus olivicens, the western jack-o'-lantern. A lot of times you think you gotta go someplace special to see animals that glow. You could just go to Lake Merritt. This is a bioluminescent mushroom, which means bioluminescent means that it produces its own light. So you could just go see this at night and it is glowing. This one right here was picked from what used to be my side of the lake, just on the base of a dying oak tree. They're not necessarily parasitic, but they're definitely right in there to kill the tree at the end, or at least make use of the good yummies that are left over from a dead tree, i.e. all that cellulose. These are most commonly seen at Lake Merritt on old oak stumps at this point. The visibility of brightness on these changes depending upon the age, and so you might have to go check a couple times before you can actually see one that's glowing. They're also famous for making a really, really nice dye. And so if you're a dye person, like dyeing yarn type of stuff, these make a beautiful violet color. All you gotta do is crush the mushroom up with a little bit of vinegar and then put your wool or your silk in there and you get a really, really nice, bright lavender color. Since David had concerns earlier, or David's mom had concerns earlier about those mushrooms that are dangerous, let's cover those right now. Amanita phalloides, i.e. the death cap, is probably one of the most notorious mushrooms in the world. It is responsible for the majority of mushroom deaths globally. It is also not ours. It's from Europe. Probably came here on cork oak or another nursery stock from Europe. This mushroom is notable because it produces not just one, but many compounds that could kill you on their own. The most notable one, though, is alpha-amanitin toxin. This toxin basically stops RNA polymerase II from working. So in a little bit more layman speak, the information that's in your DNA can no longer get translated to make proteins, which are basically the building blocks of all your cells. And so if you can't repair things by making proteins, things start breaking. And so this usually leads to death via organ failure of the liver and the kidneys. And it doesn't take much, like half a cap can kill an adult occasionally. There are protocols now for if it is known that a person has alpha amanita toxin poisoning that they can run that gives you a much, much higher success rate than just a handful of years ago. And those protocols were actually developed just out of Santa Cruz, a doctor there that's had to deal with a lot of amanita poisonings. These are mushrooms worth learning to identify. You don't have to worry about touching them. All the poisonous mushrooms in the world, they're fine to touch. It's just no going in the mouth and down the hatch. So these have white gills. The gills don't attach, so they're free. And then the cap usually will range from white to kind of this greenish metallic-y sheen. And then for an amanita, they also are erupting out of a little egg sac and have what we call a vulva at the base. And so you'll have this what we call a flaring vulva, which is basically you've got a bunch of flaring white tissue coming out of the base. And anytime you're trying to identify mushrooms, especially ones that are deadly, I would say you want to use multi-factor identification here. So you're going to be looking for the color, the vulva, the gills, and then also it has a annulus, a skirt that hangs down. But some of those can get obscured due to environmental weathering. So that's why you're always looking for multiple kind of features when identifying any mushroom, especially a deadly one. The scary thing about this is from a deadly point of view is when you ingest this, it tastes delicious, according to those that have survived. And you actually don't feel sick for quite a long period of time. So you don't start feeling sick usually till 6 to even sometimes 24 hours later. And at this point, the toxin is so far through the body that it's really, really hard to deal with. And this is why that kind of protocol from Santa Cruz now is a really, really big thing. And this is something you see a handful of deaths, unfortunately, in California from every year. Amanita muscaria is another Amanita genus mushroom. And so these are these mushrooms with white gills that kind of emerge from a little egg sac. This one is notorious in other reasons. This one doesn't necessarily kill people. Although if you live in Eastern Europe, some of your grandparents might tell you this would kill you just to keep you from touching it. These ones have psychoactive properties, but usually from people I've talked to that have ingested them, not something they ever want to experience again, like very strong kind of dissociative behaviors and things like that. These are, like, people call them the Super Mario Brothers mushroom because if you played Nintendo video games, it's the Mario Brother mushroom that you use to grow big and things like that. These are mycorrhizal associates, just like all of the Amanita mushrooms. So these ones around Lake Merritt really like the Atlas cedars that have been planted around Lake Merritt. And so we'll only get a flush of them usually every handful of years when we get a really, really big rain year. So the last 2 years, these actually have been out in good numbers around Lake Merritt, but otherwise they're usually just hiding in the soil, waiting for that good year to come about. People do eat these without the psychoactive components in there by just parboiling them. So if you boil this mushroom, it removes the ibotenic acid and the muscimol, which have the neurological effects. And then by parboiling it like 2 or 3 times, they can then take the mushroom and then cook it, and it's perfectly fine. It's safe. And this is a traditional thing that's done in Japan and some other places. A very divisive mushroom, though, too, within the mushroom community. Armillaria mela, the honey mushroom, is a notorious mushroom because Armillaria genus is probably, at least according to Guinness Book World Records, the largest organism on the planet, or one of the largest organisms on the planet. So in Eastern Oregon, from the air, you can basically see this large group of Doug firs that are not doing well. I think it's like five kilometers in diameter. And this is all one single organism. It's one single Armillaria attacking all of those mushrooms or all of those trees. We have one at Lake Merritt that is not quite 5 kilometers in diameter, but it's getting to be a kilometer from what I can tell. So I started observing this organism probably somewhere around 2007. I noticed it was killing some of the birch trees in the gardens, and it seems to have spread out and is now out by the big water fountain outside of the gardens. It has now taken down at least 2 oak trees over there. And my gut feeling is it is the same organism, that mycelium is just spreading. So these are parasitic fungi. They attack the host, and they do a pretty good job of it. They'll usually take down a tree within a handful of years. The mushrooms are edible. They get their honey name due to the sweetness of the stems. Well, some people say that the stems are very sweet if you cook them, or the stickiness of the cap is like honey. But these can be very, very large clusters at Lake Merritt. I think I've pulled up ones before that weighed somewhere on the order of 20 pounds. So a lot of biomass. Agaricus xanthodermis. This is one that Katie just saw a couple weeks ago, according to iNaturalist. This is probably the most common agaricus mushroom that we have in the grass lawns at Lake Merritt. And it's actually probably the most common agaricus we find in urban California. And so it produces these huge arcs of white mushrooms with gills that will start kind of pink and then eventually change maturity to be like chocolatey brown. If you find a young one and you rub the top, they will oxidize very strongly to turn bright yellow. And this is where it gets the name xanthodermis from. Xantho means yellow, dermis means skin. So this is the yellow-skinned agaricus. This is one that makes most people usually very sick if they try to eat it. And so this is why David Aurora calls this part of the agaricus lose your lunch bunch. And this is why learning to forage mushrooms, you should always go with somebody that knows what they're doing for a while before you start to branch out on your own. This mushroom at Lake Merritt occurs at least in 8 locations, and the biggest arc that I've seen has been approximately, I think, wow, that was almost, probably 10 meters in diameter. I mean, they get huge occasionally, especially during the big rain years or the year after, actually. We have other agaricus at the lake, the salt agaricus, Agaricus bernardii. This can be differentiated from the other one in that if you rub its top, it's not going to turn yellow on you. It also has more of like a crocodile skin-like cap to it. And if you cut it open in half, it'll oxidize pink, where the other one will oxidize a little bit of yellow. And if you were to smell this one, it smells like a tide pool. It smells nice and salty like the ocean, where Agaricus xanthodermis will smell like a chemical cleaner you'd find in a dentist's office called phenol. Agaricus augustus is also a well-known agaricus from Lake Merritt. It's called the Prince Augustus because out of the agaricus mushrooms that people forage for the table in California, this is one of the most choice ones. They get to be dinner plate size occasionally. They've got this kind of regal looking pattern, the scales coming down them, and they smell like marzipan. And they have a sweet flavor on top of kind of your normal kind of like mushroomy flavor. These like debris zones, and so any place there has been debris building up at Lake Merritt, especially woody debris mixed with lawn clippings, you'll then find this mushroom there. Leucoagaricus leucothyces, the white dapperline, is one that is also starting just to pop up right now around Lake Merritt. And this will look like an agaricus, but then when you get over and you look at the gills just never change color. And so this is why it's called leucoagaricus, leuco being the whites, and then these are basically the white agaricus. These are also edible, but pretty much nobody eats them because they are scarily close looking to an amanita mushroom that is deadly. And so most people just stay away from these. But the way to differentiate this from an amanita is if you look at the base of it, there's no vulva that's coming out of it. This is also a saprotrophic mushroom, a mushroom that eats dead stuff, where the amanitas, remember, are all mycorrhizal. They're living in association with the tree, getting a sugar cooked back from the tree, and then they're usually bringing micronutrients up for the tree. Leucoagaricus barsii, another white dapperline that we also find at Lake Merritt, very similar to its cousin there, but it's just much shaggier on the top. This is also one that not many people eat, but it is a nice choice edible. Chlorophyllum bruneum. This is an interesting one in that we can see this at Lake Merritt almost throughout the year, depending on how hard they're running the sprinklers, either in the garden or on the lawns. This can be a very, very large mushroom. I think the largest one I found is a little bit bigger than my computer that I'm working on right now. These also like debris piles. They're saprotrophs. They're also an edible species, but can be confused with another one called Chlorophyllum molbites that is known as the sickener, which has green gills instead of these white gills. But we don't really see, oddly, we don't see Chlorophyllum molbites yet in the Bay Area very frequently because we're just a little too cool. But I would guarantee you within the next 4 or 5 years, we'll start to see it at Lake Merritt as things continue to warm up during the summers. Candlomyces, the pale brittle stem. These are also one of these mushrooms that will occur just in horrendous numbers occasionally around Lake Merritt, usually where somebody cut down a tree in the last couple of years. So you'll see them popping up out of the grass, but they're really there, usually breaking down wood that's hidden beneath the grass. They'll also show up in the grass right next to the wood chip landscaping because wood chips occasionally will find their way into the grass and these will eat them. The way to identify these is that if you pick one up and basically squeeze it with your hand, it'll just fall apart on you. It just cracks right open. And that's why they're called the brittle stems. Also an edible species that some people around here do eat. Bulbolitis titibens, the yellow field cap, is one that loves the summertime at Lake Merritt, which is not a time that most mushrooms love California because it's hot. But Lake Merritt stays cool. We throw water on that grass, and these mushrooms come up in great numbers. So they're a bright yellow little thing that sticks up out of the grass. And if you were to spore print them, they have the brightest orange spore print for a mushroom of this color. They're almost kind of confusing in that regard because if you look at the gills, you're like, oh, you're definitely white spored. And then you spore print them and you're like, did not see that coming. Conocybe alpala is another one that hangs out in the grass with the yellow field cap. These are called the milky cone cap. So the way to differentiate these is these will be a tight kind of cone. They never really get bright yellow. These at one point, people feared that this entire genera was dangerous, that they had those amanita toxins that we talked about in Amanita phalloides, but further recent testing shows that they do not. Pinalus fowensii. This is the mower's mushroom because lawnmowers are probably what propagate this thing. Also very, very, very happy in the summer months and will continue on usually until about November. These mushrooms are probably have been consumed many times around Lake Merritt by people thinking they are psychedelic mushrooms because they look just like them. They're in the same genus, but all you're going to get is a little bit of extra serotonin from them and not any of those tryptoamines that you're looking for. Marasmius oretes, the fairy ring mushroom, is a mushroom that you can see even when the mushrooms aren't there. And so if you're walking around Lake Merritt and you occasionally see these giant arcs in the grass that are much greener than the rest of the grass, it is oftentimes this mushroom. And then if you're there at the right time of the year, what you'll find is the outside of that ring will all of a sudden be lined with mushrooms. And this is why it's called the fairy ring mushroom. Also an edible species, but also one people don't eat very frequently, mainly because we mainly see it in urban habitats where it's not really a good idea to eat a ton of mushrooms. Lauratomyces ceres, the chip cherries or the redheads, are very, very common throughout all the wood landscaping at Lake Merritt. This is likely not ours. This is, from what we can tell, from New Zealand or Australia, but a really, really stunning mushroom. Psilocybe elenii. So we do have probably 4 mushrooms containing the psychoactive substance psilocybin at Lake Merritt. This is one of them. This was actually described by a mycologist in Oakland, but not to steal their thunder, that name is going to get changed real soon here because a bunch of these mushrooms that were discovered on the West Coast that we'd given different names to that were in the genus Psilocybe that contain psilocybin appear to all be escapees from Australia. And so from genetic studies, these are all going to come back into Psilocybe subaeruginosa probably at the end of the year. Coprinellus myceus, the mica cap. This is the mushroom you don't want to see at the foot of your tree. Not that it's killing your tree, but it tells you the root system is dying. And so in many cases at Lake Merritt, I've seen these prop up next to the bottom of a tree. And just every time I'm out there, if I see it, I'll photograph it with iNaturalist. And now looking back, I can say, yep, saw those started coming up in 2014. And by 2018, the city came through and removed the tree because it got unstable. And so if you see these, they're usually a good indicator that something's not going well with a root system. These come up in huge numbers. They're part of a group that people call inky caps because they will do what we call deliquescence. So instead of waiting for somebody to come move their spores, what they'll do is they'll just start basically self-dissolving as their spores mature, with the intention that if somebody walks by, they can wipe them with a gooey mess of spores, and those spores now get walked someplace else. In that same group, we have the hare's foot inky cap. This one, same general strategy. It's usually not there when trees are dying. It likes the wood chips for some reason, but it's called a hare's foot because when it's first coming up, it's covered in all these small flocculations that look like a rabbit's foot. Hyphiloma fasciculata. This is probably the most strongly UV fluorescent mushroom you'll see at Lake Merritt. So if you walk around Lake Merritt with a UV light at night, you might be distressed by a lot of things, but this will be one of the things that you will enjoy seeing, is this really, really brightly fluorescent mushroom that degrades wood. Gymnopus brassicolans, the cabbage parachute. This is given this name because it smells like cabbage. If you crush it in your hands and you smell it, you're like, ooh, yeah, that's some cabbage, not doing well. Edible mushroom by some, but many people don't pass that scent. Also loves wood chips. Tubaria, there's a ton of what we call the tedious tubes, so I'm not even going to cover them, just know they exist. Sterum hirsutum, this occurs on some of our trees as they die. It's also known as the false turkey tail. It'll be these kind of nice brown shelves coming out of a tree that have kind of the zonation like a turkey tail would have. Lepista nuda, this is the bluet, a sought after edible mushroom. It likes our oak habitat, specifically the deep oak duff it needs in order to actually get fruiting bodies up, but it has a wonderful lavender color. One of these mushrooms that was rare per se in California until we started seeing it around Lake Merritt is Schizophyllum or Schizophilum, depending on how you say your Latin, amplum, and this is known as the poplar bell. And so this is a species actually known from Europe on Populus nigra, who happen to be around the lake now. And so if you check Populus nigra sticks during the winter, you will find tons of these. So not really that rare, probably just underlooked for in California. Sparabolus stellatus, the cannonball fungus. Need a mushroom that can shoot cannonballs at a light target? Here it is. These grow a lot of times on the tea trees around Lake Merritt in the kind of dead debris. And if you take one of these and you put it in a cup and you punch a hole in the cup, like cover the cup and punch a hole in the top, so there's just a little bit of light coming through, this will literally aim its spore cannon right at that and then shoot it towards it, which is kind of a really cool evolutionary adaptation, because if you're growing on dead wood in places, you probably want to try to get yourself out and away, from the rest of your cohorts who probably are already on that wood. And so it's probably a good idea to shoot towards light. And so that's probably why these are phototropic shooters. We also have scleroderma around the lake, also known as pigskin poison balls. Not bad for humans per se, but these usually do a number on dogs. I'm part of like a mushroom consultants group for vets and things, and this is one of the things we see a lot of dogs get into, unfortunately. Yes, Katie.
Hey, Damon, this is fabulous and beautiful. Do you have time to take a short pause for us to do our end of hour?
Sure.
Thank you so much, Damon, for this fabulous and just dazzling presentation of mushrooms in general and the kinds of mushrooms we might find at Lake Merritt. We're so enthralled by this presentation. It's absolutely amazing. I want to thank you for coming, and we want to thank our partners and visitors throughout the month, and invite you to come back in November for the next Lakeside Chat. Coming up in November, on November 8th, we're going to be delighted to have another iNaturalist luminary, Leslie Harris, who is the collection manager for polychaete worms at the LA County Museum in Natural History, and she's talk about the polychaetes of Lake Merritt and adjacent areas. And I don't know how many times people have asked me what various worms were that they found at Lake Merritt, or the effusion of worms swarming in the water. What could be going on? What does that mean? Leslie is going to tell us a lot about the incredible adaptations, life cycles, and beautiful structures of the polychaete worm. So this is just going to be a really exciting presentation, and I invite everyone to join us. We do Rotary Nature Center Friends Stewardship Work Days, generally on the third Saturday of the month. Details are going to be on our Facebook page. I will be going out tomorrow to do a plankton sample and water quality testing, which we continue to do as part of our regular program with community people. You're welcome to out and join in the monitoring of Lake Merritt water quality. And finally, we want to remind everyone that, of course, the Autumn Lights Festival is coming up this month. If there are any tickets left, it's a sight not to be missed. It's educational, it's beautiful, and we encourage everyone to check that out. Okay, Lakeside Chats are rebroadcast by the City of Oakland KTOP TV every week on Sundays from 6 to 7 p.m. The first 2 Sundays of the month, we'll have a collection of golden oldies from our now 47 episodes of Lakeside Chats. And then the last 2 Sundays of the month, we'll have a rebroadcast of the that month's Lakeside Chat. In this case, it will be Damon's. So we invite you to check that out and share that with people who may not have been able to attend this presentation. You can also check out the recording, which we'll have available probably in about a week or week and a half of the program, which will be on our playlist. If you enjoy what we do, please consider making a donation to Rotary Nature Center Friends. You can do this by using the donation button on our website, or by mailing a check, if you'd like to Rotary Nature Center Friends. And this will help us to provide kayak experiences, to renew our equipment, to produce our shows every month, and to improve our website presentation and so forth. And we really appreciate your feedback and support on Lakeside Chats and our programs at the lake. This is what our website looks like. You can donate by PayPal, or, as I said, by mailing a check. We want to thank the Frederick E. Hart Foundation for educational opportunity for supporting, especially our Lakeside Chats over the years. And we're hoping that this body of videos will become more and more useful as time goes on in education at all different levels in the area. I think Lake Merritt is special in what it provides for education. We want to thank all of our volunteers, our program participants, and our partners. And we want to remind everyone that we are a all-volunteer nonprofit advocating for the Rotary Nature Center in Lakeside Park as an interpretive education and science center for all the people of Oakland, and as the steward for the Lake Merritt Wildlife Refuge. We've had a wonderful program tonight, and I wanted to, before we close, make an announcement or a update about the situation with our Hank, our pelican that is a disabled rescue pelican at Lake Merritt. And we've received information from the people who take care of her that she is eating well, and that the American white pelicans that keep her company at Lake Merritt are here sometimes, but not all times, and that the hot weather has caused them to not be around for a while, but then they come back. What's really cool about this program of looking at helping to support a disabled non-wild pelican is that we're starting to learn about the individuals that come to join her and see them returning on a daily basis, on a weekly basis, and even on a yearly basis. And Rotary Nature Center Friends is looking high and low for a good naturalist, scientific presentation about the biology, life history, and conservation of American white pelicans so we can understand the wider context. So look for that in the future. And we want to thank our producer, Rob Lamone, and David Wofford and I are the co-chairs of Rotary Nature Center Friends. We thank Janice Lord Walker and Betsy, and Pat McDonald, and many RNCF volunteers throughout the month, throughout the years as we've been working towards bringing nature and people together at Lake Merritt. Thank you all for coming tonight. And now Damon has graciously made it willing to continue his presentation and to take questions.
Okay, so we also have rusts at Lake Merritt, and so these are most common right now as plants are starting to kind of die off. You'll see plants with these large kind of blistery infections all over them. So these are basidiomycetes, so related to your large mushrooms. We also have, I know a lot of people think they have to go to the tropics to see fungi that are eating insects. We've got plenty of insect eaters around the lake. So we have fly death fungus that you can find killing flies around the lake. These are interesting in that they change the neurobiology of the fly. And so when the fungus infects the fly, it basically disables the wings so they can no longer fly. The fly then gets what they call summit disease. And so it'll basically walk its way up, usually, about head height off the ground. And then it'll put its proboscis out, its little tongue-like thing, and onto the substrate. And then the fungus will actually grow down the tongue, more or less, and then attach it to whatever the substrate is, and then hold it there. And then at that point, usually right around that time, the fly actually dies finally. And then the fungus erupts from the abdomen. And that's what you're seeing here, this white fuzz. And then that spray you see on the gills, on the wings there, those are all spores shooting off. And why would you want to get up high? Now you're high enough to then rain spores down on everybody else that might be beneath you, hopefully other flies for the case of the mushroom. We also have a similar thing going on with spiders at Lake Merritt. So there's 2 oak trees that I can usually find givealulas on every springtime. And so these are basically playing a very, very similar role on spiders, infecting spiders, and the spiders will basically become hosts for all these little mini fungi. We also have the most common, basically, insect killer on the planet hanging out at Lake Merritt. If you flip over logs, you'll find these during the winter, and this is the white muscadarian fungus, and this is oftentimes infecting beetles. So you'll find beetles that are just look like they're wearing a fuzzy coat. It's the fuzzy coat of death. We also have fungal STDs at Lake Merritt, not for us, but for the ladybugs. So some of our ladybugs, if you're looking at them towards the end of summer, they'll have all of these yellow projections off the back. Those yellow projections are actually a parasitic, well, it's hard to say parasite. It's a fungus that attaches to them. It doesn't seem to change mortality of the beetles, which is interesting. And actually this one here on the 20-spot ladybug beetle, a researcher from Harvard contacted me who was studying these, and we sent him a handful of these, and this is an undescribed species of fungus. But this is a common thing that you see on ladybird or ladybug, lady beetles across North America. All the different species of beetles appear to have different species potentially. A really cool mushroom that sometimes you can smell but not find at Lake Merritt is the candy cap. And so it's hard to Lake Merritt because there's something else that smells just like this, which is the acacia blooms will do this too. It smells like maple syrup. And so this is a mushroom that people use actually as kind of a sweetener. And so they'll get used for making cookies. I used to use it to make beer with because it's a way to add sweetness to beer without having a fermentable sugar there. And there's some really cool organic chemistry that goes on there, but we won't get into that right now. We've actually got both of California's candy caps. This is the Rufus candy cap, the other version of this, basically. And this one was really interesting because years ago, when I first got interested in sequencing mushrooms, I was starting a project at Lake Merritt where I was hoping to do barcoding at the lake more frequently than we ended up doing. There were some snafus, let's just say, in the process of working with large entities, and we only got a couple things out the door. But we started sequencing some of these mushrooms, and that Rufus candy cap was one of the first ones we sequenced. And kind of surprisingly, nobody had ever generated a sequence of that before. So we didn't, at that point, we had like a sequence that didn't match anything, which means A, this is maybe a new species, or nobody's actually sequenced what we call a holotype in biology. So that kind of the thing that everything else is named after in a species. And so we're still waiting for that to basically happen for that one. In general, this is the process that we were using at Barcode the Lake. We would document with iNaturalist, take a voucher of that organism. So I've got something to go back to, do microscopy on it, extract DNA, amplify out just a small portion of the genome, confirm that that happened, and then sequence that. And in that sequence, we would then use to determine, is this what we think it is? Has this ever been sequenced before? How does this compare to other organisms out there? And since Barcode the Lake, I got some stumbling blocks. I moved on to helping a group that now just looks at all of California, trying to understand fungal diversity. And so as of this morning, we've got about 10,000 specimens collected in the last year and a half from across California. So these have in-depth vouchering done, lots of field tests, chemical, UV, things like that. Physical specimens that now reside at California State East Bay, which then means we can do sequencing on them. So we've been sequencing these like crazy. Well, I haven't, but Hart Singer has been doing this at Fundus. And we've got, I think now probably somewhere around 7,000 of these have been sequenced. And the crazy thing is that from the first year of looking across California, not too surprisingly, a whole bunch of these were relatively novel. So depending upon how you use the number or what you mean by novel, it's somewhere between 10 and 35% of these were basically new. Which means that there's a whole bunch that we don't know about fungi. And this is why we're running this project for at least another 5 years. We can continue the funding. And so the hope is this. We're basically seeing new species so quick right now through the use of basically DNA sequencing, microscopy, and other tools that we kind of all just generally decided that it's not worth it to stop and name species. It'd be way better just to have known that you've seen everything first and then go back and name stuff. It's a way better way to do that because then you can actually apply appropriate names. It's like if you were to go explore a new country and you started naming things tallest mountain, and the next day you saw another taller mountain, you're like, oh, maybe I should rename that first one. And so I think hopefully in California by 2028, we'll have enough collections and enough sequence that we'll at least know the general landscape of all of the mushroom diversity in California. And then some academic folks will probably sit down and start putting names on stuff. So that's basically the whole talk. I'll open it up for questions. It looked like there was a question that just came in the chat. Let me just roll up to that. Are there mushrooms that have symbiotic mutual benefits relationships? Oh, it just moved on me. With other species. Yeah, a good portion of these mushrooms at Lake Merritt are actually have symbiotic relationships. So any of those ones that I noted were mycorrhizal. So things like the Amanita mushrooms, those are all in symbiotic relationships with trees, where they're basically gathering up micronutrients for the tree, and the tree in return is usually kicking back sugar. And then also, besides just micronutrients, they're also really expanding the overall surface area of the root system, which means you can then take up water much more effectively as well.
There's a question.
Amanita virosa, we do not have in west of the Rockies. And so we have not seen that at Lake Merritt or in California. In California, we really only have two deadly amanitas. It's Amanita phalloides, which isn't ours per se, and Amanita ocreata, which is ours, which is the western destroying angel that associates with coast live oak mushrooms. It's really only frequent, though, in the spring season. So
there's a question from Lila. Do you know of any application of the fly death fungus in attacking cancer cells in humans?
I have not heard of an application of that one against cancer cells in humans. There's other mushrooms that are used potentially for cancer. I mean, actually, death cap, those toxins are really good for killing cancer. You just need to make sure you deliver it just to the cancer cells. And so people can do this through kind of immunotherapy approaches. But as far as I know, I haven't heard of any of the fly death fungus being applied for cancer. And it's mainly because most of the active components there are neurology focused compounds. So you find a lot of like methamphetamine class compounds in fly death fungus and things like that, things that activate neurons. Let's see here, symbiotics. Have you seen? Okay. All right. Next one from Anthony. I live near a company in upstate New York named Ecovative that uses mycelium to grow products ranging from leather-like textiles to sustainable packaging to high-performance foams. Ecovative has been widely recognized for technical environmental contributions to compost packaging. They have a division that has developed. Yep. Yeah. So, I mean, this is one of the interesting things right now is that within kind of industry, you're seeing mushrooms become a really hot thing because they produce a lot of very interesting material compounds. And so there's Ecovative is one of the big ones because they have done a really good job at doing the R&D and patenting it. But you see people growing them as far as making substitutes for leather, making packaging, things like this, making protein-based like food substitutes. And I think this will be a thing you'll see continue to explode. And we have a number of businesses doing similar stuff in the Bay Area, most of them on Ecovative patents, though. Andrew still asks, hey, any boletes? There's a handful of boletes that will show up at Lake Merritt. So I have found Boletus edulis and Boletus barrousii at Lake Merritt before. And those will be on the Atlas cedars. Can you say just a bit more about mushrooms being more like animals and plants? So they're more like animals and plants because of the way that they feed. They are heterotrophs, so they eat other things. They can't produce their own energy like plants do by collecting sunlight and carbon dioxide and doing that crazy thing called photosynthesis. Also, just from a genetics point of view, if we look at them, they are much more closely related to us than plants. And so if we use that kind of phylogenetics approach that I kind of did a surface dive on, that would also show that they are much, much closer to us than to the plants. Why are morels so hard to cultivate commercially? It's mainly because they have a very funky lifestyle that isn't as easy as a lot of mushrooms people are used to cultivating. Most of the mushrooms that have traditionally been cultivated are saprotrophic mushrooms that live on dead stuff, which makes it really easy because you just give them a pile of dead stuff and they will be happy to grow and fruit on it. However, though, a lot of these things that are mycorrhizal are tough, like chanterelles, like boletes. But morels operate in even a harder space because like a chanterelle or a bolete, I could grow the tree and then get the mushroom. In fact, people do this with truffles and things like that. But morels are a really interesting one because we think some of these morels are endophytes, which means they actually live inside of the tree and protect the tree from bacteria and other fungal infections. And only when the tree host starts to die does the organism known as the morel go, uh-oh, I got to get out of here, and put up that fruiting body, the morel that we are familiar with. And so that makes a lot of the morels hard to cultivate. There's a small subsection of them, though, that are saprotrophic. And so people have just within the last well, I mean, the first paper was actually in the 70s, but the first major, like, large scale cultivation of this has really only started in the last decade. And so a lot of those morels that you find at Costco are actually cultivated morels from China right now, fetching the same price that fire morels used to get. So somebody made a lot of money there for a couple of years. As a beekeeper, I've been reading about research looking into using certain fungi for treating varroa mites instead of chemicals. Yes, bees are known to go get metabolites from fungi in order to fight off infections, including varroa mites. And so famously right now, or at least maybe overly hyped to some degree, a lot of this has been basically fungal extracts coming from a lot of the kind of shelf mushrooms like Ganoderma and things like that are being given to beekeepers as a way to help their bees basically deal with varroa mites. Even a better solution would be just let a tree next to you decay. The bees will find the fungus in there and get the metabolites from it. A lot of this is that a lot of our bees are being used in areas where they just don't have access to the mycelium anymore. Remember, most insects and other animals, they are very good at seeking out the compounds they need to deal with parasites and things, but we've a lot of times removed them from the environment because we don't like to see stuff decay. You mentioned earlier in your presentation that there is contrary disagreement about Amanita muscaria. The disagreement within the mycological community is whether or not it should be touted as an edible. One of our actually well-known Oakland mycologist who is an Amanita expert is like, nobody should be telling anybody to eat Amanitas, period. But then you have David Aurora on the other side. Remember, they got kind of rebirthed mycology for non-academics, who's like, no, this should be like one of the first things you eat because it's easy to identify. And it also gets you in the habitat of thinking about, how do I prepare food to make it safe? Because a lot of the foods we eat, right, have been prepared to make them safe. I mean, a lot of the grains we eat have been prepared in such a way to make them safe that if we didn't do that, we would get sick. And so that's the argument there, is mainly on the edibility and telling people that it is an edible mushroom. The Linnaean Society of London had a program earlier today about mold. Mushrooms and medicine, our lifelong relationship with fungi. Cool. Yeah, it's a blossoming area right now. I mean, everybody should be aware of fungi as medicine ever since Fleming found some penicillin in a dish, prior to World War II, because it became basically the breakthrough, I would say, for having finally tools to fight bacteria effectively with. It's really expanded in the last couple of years as people have gone full bore, basically bioprospecting in fungi for new compounds. You have to remember, a lot of these compounds do things that are very novel that we haven't been able to find in other places. I know people think about antifungals from mushrooms mainly being things like penicillin, but if you think about some of the biggest blockbuster drugs in chemotherapy like Taxol. Taxol is derived from a fungus. It was originally thought that it was from the Pacific yew tree, and they were like, this is great, we've got this blockbuster drug, but how are we going to grow enough yew trees? And then in the process, somebody realized it wasn't the yew tree, it was the endophytic fungi living inside the tree that was producing the Taxol. And that fungus is a lot easier to produce. And so that's why we have Taxol as a chemotherapy drug. Also, cyclosporin, one of these compounds that you would use if you ever had an organ transplant, is also fungally derived from one of those kind of scary looking fungi that attack other organisms. So there's a whole host of things basically rolling out of this bioprospecting pipeline right now for useful medicinal compounds out of the fungal kingdom. Any other questions, or did I miss anybody's question in the chat there? I think we got most of them. How is CRISPR related to fungi? Interesting. So CRISPR is derived, CRISPR, if you're not familiar, it is a genetic editing tool that's kind of gotten a lot of fanfare recently, mainly because it's really easy to use, really cheap to use. It was derived originally from bacteria, from Streptococcus. But you could deploy it in any organism. So people are using CRISPR to modify fungi. So you could literally go in, tell this more or less protein complex where to go cut in a genome, it'll go cut, and then you could do stuff with that cut. You could put a new gene in there, you could do a cut someplace else and remove a gene. So it's very useful. Fungi will deliver us new mechanisms on maybe not the usability of CRISPR, but similar. There's one called Starship, named after the Starship Enterprise, that is really good at picking up very, very large genomic chunks, like 200 kbs, and moving them around. Olivia asks, are there any potential non-native fungi found associating with non-native plants in the botanical garden or elsewhere around the lake? That's a really good question. With the eucalyptus is where we will find, it's easiest to find those non-native mycorrhizal fungi. So we do have a truffle that shows up at Lake Merritt that I did not cover in the talk that is associated with the eucalyptus trees. It also has a Lacaria mushroom, which is a gilled mushroom that's kind of pink color that will also show up with the eucalyptus, which is also non-native but associated with a non-native plant. And I mean, the Aminophyloides, it's also the ones at Lake Merritt are associating not with one of our oaks. The last time I saw it there, it was on some sort of weird hybrid home oak from either the UK or Northern Europe. So even that one's there. What's your favorite mushroom to eat? Man, that's a tough one. I really like yellow feet chanterelle, mainly because I can pick a whole bunch of them in a season and they preserve really well. So if you dehydrate them and kind of keep them in a jar, they get kind of cheese-like smelling over time. And if you add them to dishes, they just add a really, really big pop. And they're also one of the cleanest mushrooms to pick, so I don't have to spend hours cleaning them when I get home. I just throw them all in a dehydrator and I'm done. Miriam, possibly another reason it's hard to grow some mushrooms is bacteria and viruses that live symbiotically with the fungi. Yes. Especially with a lot of these mycorrhizal fungi, my guess is they need other environmental factors like bacteria or signals from the host tree. Hey, it's that time of year again, you should think about fruiting. In fact, the few boletes that are being grown right now in Thailand, they can only be grown if you grow a bunch of the mycelium and then you cap them with soil. And that soil tells me there's the bacterial component or maybe a viral component that's triggering the fruiting. Are mushrooms attacking the giant redwood in front of the RNC? Nope. Redwoods are pretty much never going to be taken down by mushroom. They have evolved to deal with fungi so well that barely anything breaks them down, even when a tree falls. They don't have any mycorrhizal associates. Some plants were like, hey, you know what? I'm going to partner with fungi to make my life easier. Redwoods were like, it's all right. I got my bases covered. I don't need any of you. And so I don't think it's a fungus taking down the redwood near the RNC. In fact, I haven't noticed that it's in bad shape, but maybe next time I'm there, I will go take a look. Could be salt intrusion. Due to, you know, things are going up. Are there aquatic mushrooms? Yes, there are definitely aquatic mushrooms. The most famous one is from the Rogue River in Oregon, just across our border. It's Sathoralia aquatica, which is the only well-documented mushroom that fruits underwater. And so if you're snorkeling in the Rogue, do this in the summer. You will occasionally find what looks like a mushroom, because it is, coming up off of wood in the bottom of the river, and it will let its spores go into the water column apparently. But there are lots of other aquatic fungi. So if you look at any aquatic plant and you were to take it, basically sterilize some of the tissue and then put it in a medium that you could grow fungi from, majority of the time you'll see fungi come out of any of those aquatic plants. So if you do that with like widgeon grass that we find all the time, or eelgrass, in kind of estuary systems, you'll find that they have tons of fungal endophytes, probably. Thanks, Anthony. Yep, and if anybody has any further questions, you can find me pretty easily on social media platforms just by typing in Damon Tighe. Turns out I'm the only guy, at least in North America right now, with that name, so I'm pretty easy to find. There's one bloke out in the UK with my name, but we know each other, so we sort each other's mail, so it's all good. But yeah, if you've got any other questions, you can find me Instagram, Facebook, iNaturalist, and just at me on one of those, and I'd be happy to answer your mushroom questions for you.
Thank you so much, Damon. And yes, please, I want everybody give him a hand. It was just a wonderful presentation. And we will be sending out a post-chat email to everyone with some of the links in the that were in the chat and some of the great questions. This is really an awesome discussion. And thank you so much for coming. And I hope to see you again.
Hopefully at the lake.
Yes, at the lake. That would be awesome. I'm going to be there tomorrow doing water quality and plankton just to see what's there. Okay, everybody, thank you so much. And we'll see you next time.